Well, hello, we are here on a spotlight interview with Noah Langenfeld, who received dual undergraduate degrees in biochemistry and biology from the University of Wisconsin, Stevens Point in 2020. He's currently a PhD candidate in plant science at the Crop Physiology Laboratory at Utah State University in Logan, Utah. His focus is on using recirculating hydroponic systems to develop sustainable fertilization programs and recycle nutrients for life support in future space missions. He has been the first author on 5 peer-reviewed articles so far during his PhD, and 2 of those articles are lab protocol papers. Ultra running, hiking, and aviating are his desired pastimes when he's not working to support our future food supplies. I am Gabriel Gasque, Head of Outreach at Protocols.io, and today we will be discussing with Noah a lab protocol paper entitled Germination and Seedling Establishment for Hydroponics, the Benefit of Slant Boards. So with that, let's kick it off. And Noah, can you briefly explain what the protocol is about, how to use it, and why it is relevant?
Yeah, absolutely. And yeah, thanks again, Gabriel, for this invitation for this spotlight interview. So the protocol is mainly designed at germinating seeds in the absence of any solid substrate. So not having any soil, no soilless media, specifically for eventually transplanting into liquid hydroponics. So one of the things we've constantly found in our research is that if we try and start seeds in soil and later transplant them into hydroponics, they go through a lot of transplant shock, and we're not able to get off all of that soil, which can lead to contamination and bacterial issues when we put them in our deep flow hydroponic systems. So one of the things we wanted to try and develop and refine was a method or a protocol for ensuring that we can get clean seedlings started quickly, and for later transplanting into our hydroponic systems. This was kind of further stemmed by, we would give a lot of tours at our laboratory, and people would always ask, like, how do you start your seeds when it's just water there? Seeds can't really germinate in water. So that's one of the things we really wanted to kind of highlight and be able to show other people and share with other researchers.
But were there any previous protocols on how to do it without a solid substrate or without a soil? And if so, what was the motivation to optimize this particular protocol?
Yeah, so our protocol is based on one, I think, back from the 1970s. It was a really old paper in kind of an obscure journal. I'm using this slant board method, mainly just kind of using it as a tool to study seed germination itself. So we wanted to kind of take that method and kind of bring it to a more, I guess, modern era through protocols.io and PLOS ONE to get it out there and kind of expand it from simple seed germination studies to actually be able to use it as a tool for other larger research projects in liquid hydroponics.
I see. And so once you optimized and brought this protocol up to date and expanded it in scope, so what was the motivation to have this protocol peer-reviewed and eventually published as a formal paper in PLOS ONE?
Yeah, so we really like the peer review process at our laboratory, mainly because it gives us feedback on things that we might not have initially thought of when we're writing something up or doing something. Like, we're kind of all plant scientists that work in the same lab, and we know all the tricks and everything, but if we write something up, it can be pretty easy to leave out important details that other peer reviewers are able to pick up on and say, hey, why didn't you discuss this? Or, hey, you need to expand on this a little bit more. So that's why we really try and go through that peer review process to be able to put things out there so we can gather feedback from the research community and potential other growers and other people out in industry that are going to be able to use this protocol and give us advice on how we can improve it.
And can you elaborate on the feedback you received from your peer reviewers and how they benefited your paper and what changed in the protocol in response to that criticism or feedback?
Yeah, so one of the main things that we kind of got back from the peer review process is they were slightly confused on the type of water that we were using to germinate our seeds. Was it water? Was it fertilizer? Was it some kind of dilute solution? What was that? And then also kind of how we maintain that in our box or our reservoir throughout the germination process. So we always knew what we kind of had to do on the back end, but kind of seeing that feedback and then understanding that, okay, we actually do need to discuss this in further detail. So we ended up adding a couple paragraphs to the peer-reviewed article. And then for the protocol, we added a couple steps, just kind of clarifying things, saying you need to use this type of water, these exact measurements of kind of depth of things to make sure that the seeds stay, they don't get desiccated throughout the process. So that really helped kind of improve the overall protocol and peer-reviewed article as a whole, I think.
And what was the motivation or the reason behind you making public the peer review history? Because this option is offered to all authors, but not everyone takes it.
Sure, yeah. We submit to multiple different journals, and some of them have that option, some of them don't have that option. We typically always choose to publish the peer review history. We kind of see it as kind of a transparency factor, how the paper went through review, especially in more kind of detailed papers, things particularly like this, to kind of see, or allow people to see like the evolution of the protocol and the paper itself, and to be able to understand that this wasn't just kind of a one and done thing, that this was kind of a work in progress over multiple months during that peer review process. So we have no qualms with publishing that peer review history.
And now I would like to move to like more technical questions about the protocol itself.
Sure.
So as you were updating this old protocol to make it suitable for your needs and for the field in general, was there anything that you tried but didn't work and didn't make it to the final version of the protocol?
There was mainly on the engineering side of the system for germinating the seeds. We tried a couple different materials. We tried a couple of different, I guess, angles of how we were going to angle the seeds to get the kind of the best germination percentage. The original protocol that we had based all this work off of had kind of just said, this is the angle you need to do, and these are the things you need to use. It kind of didn't really give any type of range or anything. So we kind of played around with that a little bit. Seeds need to be more slanted or less slanted. What kind of materials can we use? Can we use glass? Can we use plastic? Can we use wood? So initially we did play around with that, and then kind of settled on the final things that ended up in the peer-reviewed article. Just over multiple testings, the things that worked for us were the easiest to clean, easiest to maintain, and gave us the most kind of disease-free seeds.
And while you use this protocol, have you had to troubleshoot it? Are there any steps that are tricky and that need sometimes a little bit extra work or tweak here and there?
I guess the trickiest thing for us has been making sure that we actually follow our own protocol. Sometimes we'll get a little bit lazy in some of the steps, or we'll let things kind of go too long. That seems to be when problems arise. So I guess if we just kind of take our own advice and actually do things as we wrote them down, that seems to be the best option for us. One of the big things in kind of our expected results section was kind of how long you leave the seeds on the germination board. There becomes a point where if you leave them on too long, they just kind of sit there and are no longer growing. They're not going through any exponential growth phase. So sometimes things come up, we forget about things for a while or for a couple of days, and we leave them on there too long, and that ends up being problems. So paying attention to those timing things, I think, is the most important part in the protocol that people need to be aware of. So,
and then can you elaborate, in addition, to the timing, what are the most critical steps in your protocol? What are those where like users shouldn't be messing around?
Right. I think the most critical step is when we have our germination board, and then we're submerging it, the bottom of it, in this reservoir to keep it hydrated. Ensuring that tap water is used in that reservoir. That's very important. You don't want to be using any type of nutrient solution or any type of nutrients in there at all, because that's going to lead to algal growth, which is going to just consume the entire container. It's going to get on the seeds and then get later transplanted into your main systems, which you don't want. So sticking to that and only using tap water, no nutrients, that's one of the most critical parts. And then related to that, making sure that the water level kind of is in contact with the board and the germination paper throughout the process. If you let it dry out for just 30 minutes or an hour, your seedlings are going to be done. So kind of paying attention to those time things and making sure that the seeds stay hydrated with tap water is, I think, the most important part of the protocol.
And you mentioned that even for you, it is important to really stick to the protocol and follow it. And when you slack is when problems arise.
Exactly.
But have you seen any room for optimization, or have you optimized the protocol even further since publication? And if you have, why and how?
Yeah, one of the things that we kind of see for optimization is kind of the temperature, I guess, idea. What kind of temperatures do these seeds need to be germinated at? Is there kind of one temperature that might fit all, or do some seeds need to be germinated maybe at a cooler temperature, things such as spinach, or a warmer temperature, things such as tomato and peppers? So we kind of just have our system in kind of one central location. So we're not really playing around with temperature yet, but we've seen through the literature and kind of just our own experiences that the temperature does matter in seed germination, specifically how fast they're able to germinate kind of off the gate there. So that's one of the things we're kind of looking at potentially optimizing in the future, is kind of controlling temperature based on what individual species we're working with.
And now, since we have discussed now the technical part of the protocol, I would like to move back again and have a big picture, and would like you to tell us what has been the significance of specifically this paper, but in general your lab protocols papers for your career.
Yeah, so one of the most significant things I think is allowing myself and others at our laboratory to be able to forge connections with other researchers that may have been working on a similar project that we either weren't aware of, or we're having maybe trouble with that project, and we're able to kind of help them and guide them using the techniques that we've developed here. That really then kind of has sometimes led to both friendships and other kind of working relationships that we're able to work together on larger projects and kind of share information, share data, share techniques back and forth. So really kind of collaborating throughout the plant science space is what's really been a big benefit of these protocols in general.
And it was triggered by the publishing of the protocols.
Yeah. So without that, people were kind of just, I guess, as lost as we were in kind of how to do some of these things. But we were able to kind of dig through the old literature, pull these things back up and be able to kind of modernize them, optimize them, and be able to give them to modern researchers.
And I noticed that for both of your lab protocols papers, you are the first author, but you're also the corresponding author. Is that something that usually happens in your lab, or is that specifically for these protocols, and how you and your team made that decision?
It's pretty typical in our lab. We have a rather large lab, so typically the first author ends up being the corresponding author, just so they're kind of able to manage inquiries and manage any questions that may arise, as opposed to just having one central person for the litany of papers that we publish within a given year. It becomes too much for one person to handle. Okay.
These are the questions that I had for you. I would like to know if there's anything else that we haven't discussed and you would like to add to this interview.
I guess just the main thing is that I really appreciate the collaboration between the protocols.io and PLOS ONE. I probably wouldn't have published both of these protocols had it not been for that collaboration and kind of that advertising and the ability to get those methods out there. That's one of the things that I a lot of times will struggle with peer-reviewed articles, is really understanding the methods and knowing how they can help our lab as a whole. So if I'm able to help the research community to be able to better understand those methods, it becomes a win-win situation for everyone.
Well, Noah, thank you so much for your time. It's been really a pleasure to talk to you, to discuss in detail the paper and its impact on your career. So with that, I will bid farewell.
Awesome. Thank you, Gabriel. Appreciate the time.